Hello students. Hi. After a pretty long time, I'm just starting with a new topic, that is types of embryo sacs, very much in your syllabus. Let us start with types of embryo sacs. Types of embryo sacs. So one should know first what is the actual normal type of an embryo sac, which we encountered in Polygonum, and we have already studied its development. That is megasporogenesis. After megasporogenesis, one of the megaspores, that's towards the callosal end, develops into a single embryo sac, or what we call as a female gametophyte, and it bears a single nucleus which is haploid. It undergoes mitotic division, and the two nuclei are pushed towards two poles where they divide twice mitotically, giving rise to in all 8 nuclei. And 8 nuclei assemble themselves, get organized in such a way that 3 form the egg apparatus towards the micropylar end, 3 antipodal towards the callosal end, and the 2 from the 2 poles come and lie in the center, which are known as the polar nuclei. They are haploid. They may fuse to give rise to a diploid secondary nucleus, or they wait for the male gamete to come and fertilize them and go in for a triple fusion. On the basis of number of megaspores involved in the development of embryo sacs, three different types of embryo sacs are encountered in angiosperms. These are monosporic, bisporic, and tetrasporic. Monosporic, where only one embryo sac participates in the formation of embryo sac. Bisporic, where two of the megaspores participate in the formation of embryo sac. And tetrasporic, where all the four participate in the formation of embryo sac. Monosporic, where only one megaspore participates in the formation of embryo sac, have two types of embryo sacs: 8 nucleate, or we also call it polygonum type; 4 nucleate, or oinothera type. Similarly, bisporic, where two megaspores are participating in the formation of embryo sac, we call it as allium type and endymion type. Then tetrasporic, likewise, where the 4 of the megaspores are participating in the formation of embryo sac: Adoxa type, Plumbago type, Pania type, Pepromia type, Drusa type, Fritillaria type, and Plumbagella type. Let us first describe the monosporic type. This type of embryo sac develops from the callosal megaspore. Its nucleus divides twice to form 8 nuclei. This type is generally referred to as normal type of embryo sac, or also we call it as polygonum type because it was first observed in polygonum. Please observe the diagrams. Again monosporic, but a 4 nucleate embryo sac, also called as monosporic, 4 nucleate, or oinothera type of embryo sac because it was first encountered in oinothera, and mostly it's a characteristic feature of onagraceae. This type of embryo sac usually develops from the micropylar megaspore. The megaspore nucleus divides twice and forms only 4 nuclei. Out of these, 3 organize into egg apparatus, and the fourth functions as a polar nuclei. There are no antipodal cells. Here, 2 megaspores participate in the formation of embryo sac. The bisporic embryo sac develops from one of the two dyads formed as a result of first meiotic division of the megaspore mother cell. One of the dyads degenerates, both the nuclei of the functional dyad take part in the formation of embryo sac. Each nucleus undergoes 2 mitotic divisions, and as such the mature embryo sac is 8 nuclei. The 8 nuclei are organized into antipodals, egg apparatus, and polar nuclei as in polygonum type of embryo sac. In this type embryo sac, the 4 nuclei derived from one megaspore nucleus are genetically different from the other 4 derived from the second megaspore nucleus. It is also of two types. It's of two types. The bisporic embryo sacs are of two types: allium type and endymion type. If the callosal end diet participates in the formation of the embryo sac, we call it as allium type. If it is the micropylar diet which participates in the formation of embryo sac, it's called endymion type. Please observe the diagrams. Now, tetrasporic embryo sacs, where 4 of the megaspores participate in the formation of embryo sac. Meiotic division of the megaspore mother cell is not accompanied by cytokinesis. So, in meiosis, 4 nuclei are formed which are haploid. Since there are 4 nuclei inside one megaspore mother cell, we call it as cenomegaspore. All the 4 nuclei of cenomegaspore participate in the formation of embryo sac, genetically more heterogeneous than bisporic type of embryo sac. Tetrasporic embryo sacs are further divided on the basis of position of haploid nuclei in the cenomegaspore. the number of times these nuclei divide, and the organization of nuclei in the mature embryo sac. Tetrasporic embryo sacs are further divided into two groups. One where the no nuclear fusion occurs, where the nuclear fusion occurs. Where no nuclear fusion occurs, there are types: Plumbago type, Adoxa type, Peperomia type, Paenia type, Drusa type. And where the nuclear fusion occurs, we have Fritillaria type and Plambigilla type. Now we go to Plumbago type. Observe the diagram properly. The megaspore nuclei arrange themselves in a cross-like manner. One lies at the micropylar end and the other lies at the callosal end. The two other nuclei present at each side of the embryo sac. Each nucleus divides once. Thus pairs of four nuclei are formed. One nucleus from each pair migrates to the center. They fuse to form tetraploid secondary nucleus. The nucleus at micropylar end forms the egg cell. The rest of the three nuclei degenerate. There are no antipodal cells and synergids in all. It's an 8-nucleate embryo sac. The second one, where no nuclear division takes place, the Adoxa type, earlier known as Lilium type. It's characterized by all four megaspore nuclei undergoing just one more division to form 8-nucleate embryo sac, having a normal egg apparatus, 3 antipodal cells, and 2 polar nuclei. It was described for the first time by Johnson in Adoxa species. Now the second one is Peperomia type. The 4 haploid nuclei of Cenomegaspore undergo 2 successive mitotic divisions, forming 16 nuclei. Of these, 2 nuclei at the micropylar end form egg and synergid, 8 fuse in the center to form a polar nucleus, and the remaining 6, which are cut off by membranes at the callosal end, may be compared with antipodals. Thus, the egg apparatus of Peperomia type is characterized by a single synergid. This type of embryo sac occurs in members like Peperomia. The Paenia type, the 4 haploid nuclei of the Cenomegaspore undergo 2 successive mitotic divisions, forming 16 nuclei. But at maturity, Paenia type of embryo sac has an egg apparatus and 4 nuclei, polar nuclei. There are no antipodals. They degenerate. The remaining 3 group of nuclei, 1 calyzal and 2 lateral, degenerate at maturity. Highly polyploid 5X primary endosperm nucleus is formed after double fertilization. Now the Drusa type, which is 16 nucleate embryo sac, first observed by Hackinson in 1923 in Drusa, belonging to Umbelliferae. Here, children, you will just observe the diagrams. Megaspore, as a result of megaspore mother cell, as a result of meiosis, forms 4 nuclei, 1 towards the micropylar end, 3 towards the callosal end move. And you can see here they undergo first mitotic division, giving rise to 2 and 6. Then another mitotic division, 4 and 12. And they get organized into 3 egg apparatus, 3 organized to give rise to egg apparatus, 1 egg and 2 synergids, 2 polar nuclei, and 11 antipodal cells. The tetrasporic embryo sac, which are formed as a result of new nuclear fusion takes place. The first one is Fritillaria type. The 4 haploid nuclei of the Cenomegaspore arrange themselves in 2 groups, 3 at the callosal end in the form of a triploid nucleus, they fuse, and 1 haploid at the micropylar end. The triploid callosal as well as the haploid micropylar nucleus undergoes 2 mitotic divisions, and as a result 4 triploid nuclei are formed at the callosal end and 4 haploid at the micropylar end. In mature embryo sac, 3 haploid nuclei organize into egg apparatus, 3 triploid into antipodals, and remaining 1 haploid and 1 triploid nuclei move to the center where they fuse to form a tetraploid polar nucleus. The second type, Plambigilla type, observed in Plambigilla. The initial development is similar to Fritillaria type triploid nucleus at the callosal end and haploid at the micropylar end. Each of these nuclei undergo a single mitotic division and form 2 groups of 2 nuclei each. 1 haploid nucleus from the callosal end fuse at the center of the sac to form a tetraploid polar nucleus. 1 haploid nucleus at the micropylar end forms the egg, and 1 triploid nucleus at the callosal end forms a single antipodal. There are no synergids. Thus, in mature embryo sac of the Plambigilla type, the egg apparatus consists of a single cell which represents the egg. With this, we've finished the types of embryo sacs encountered in angiosperms. I would like to thank Shraddha Chavan and Vishal, my 2 students, who helped me with the diagrams, and a big thank you to all of you for patiently listening. Please Like it, subscribe it, and share it. Thank you.